David, I was looking forward to Montreal in July, but alas, I have to wait until the next time. So I'll get started. Today I'm going to talk to you about potpourri, if you will, of genomics problems. And why did I choose these problems? Because I think the questions are interesting, and also equally interesting are the mathematics underlying them that were used to answer the questions. So the talk will be in two parts. In the first part, I'll talk about comparative genomics, both interspecies and intraspecies. And the second part will be on what I call a big data genomics. Of course, comparative genomics is a huge field that stalwarts like David Sankoff and Pavel Pevsner and Tandy Warno and all the big names have contributed big time to this field. I will talk about a small problem from this space and using the lens of clusters. So here are two species. This is interspecies, and these are homologous genes. And we are looking for genes that sit together in these two species. And all the boxes here you see are clusters, and you immediately see a structure or an architecture. The red and the green overlap, and they're contained in the blue, the gray is contained in the white, and so on. So there is a complex architecture to this problem, and the immediate question that comes to mind is, let me just get my laser pointer out. What is the complexity of the problem? As you know, in computer science, even simple sounding problems like the problem of small phylogeny that David Sankoff posed isn't be hard. Of course, there are a bunch of questions around breakpoints where there are polynomial time solutions. So what is the complexity of this problem? It turns out the complexity of this is linear, not just polynomial, it is linear. What it means is that in the time it takes you to read your input, you have discovered all the clusters in their entire architecture as shown here. How is that possible? That's possible by using a structure called PQ trees, which goes back to 1970s. It's a combination of PQ trees and a very clever algorithm, Uno-Yaguira algorithm, that gives this linear time algorithm to this. And of course, you can now discover, let me just turn this off here. So you can discover clusters which have structures of this kind. So this is between human and rat on 10 segments. You can also actually count the number of such clusters. And the count is a fair measure of evolutionary distance between species rather than just the naive collection of patterns. To motivate the next problem, let's look at three clusters. Of course, one of the characteristics is the number of genes involved. And computing statistical significance, or z-score, is an important problem. And you immediately see that you need to do some kind of scoring of this architecture that you are seeing here. So how do you compare the structures, or how do you make that evaluation? So we pose the problem as follows, is that from all possible permutations of the genes, how many of them preserve the same architecture? So how do you solve this problem? You could enumerate all the permutations and check for the occurrence of your structure that you're seeing. Can we do better? Yes. We give a formulaic solution to this problem. What it means is that you don't have to solve this problem, or you don't have to enumerate the gene permutations. You just need to evaluate this, a recursive function, nevertheless. And it turned out that this also solved a classical mathematical problem, that of counting the number of simple permutations. And this is the first such explicit formula. So now you have these two characteristics of the clusters. You have the number of the genes, and you have a measure of the scores. And now you can use, measure of the architecture of these clusters, and now you can feed it your favorite z-score or statistical significance calculation and do the tasks that you want to do, for example, phylogeny and so on. And it also turned out, surprisingly, that it could be used in computer security, in malware phylogeny, in malware detection. So it was very interesting that a problem that we solved in genomics had a solution or could solve a problem in computer security. In fact, there were some companies formed around this, but I will not get into that. But if this line of research has intrigued you, there is a lot of space for you to contribute. For example, I talked about entities lying on a linear string, like your genome or maybe the computer code, but your input could lie on a grid or even on a network. And then you could be looking at problems of network motifs or network alignments. And then the question is, can you invoke constructs and get such clean and tight solutions? Now I move to the next problem. Many of you may know that we worked with National Geographic on a project called the Genographic, where the consortium gathered data from around the globe and uncovered stories. Data is genomic data, and uncovered stories of movement of people, local stories, global stories, hyperlocal stories, and so on. But today, I'm not going to speak to any of those stories. Instead, I will pursue a quaint question. So this was one of the results that came out initially from the Genographic project. What you are seeing is the common history of some 600 genomes of sub-Saharan origin. And you see that the common history, the structure is very clean. It's a tree. So how come? And that's because it is tracking the history of the mitochondrial DNA. Mitochondrial DNA is something that you inherit only from your mother, and this structure turns out to be a tree because of that. But what if we track the history of a recombining segment? What does the history look like? And this is a simulation of that. And the leaf nodes here are blue, green, and yellow. These are three different populations, and you track the history until you reach a single node, like a root node here. The question that can come to your mind is, does it converge to a single node? And it's a good question, and the answer is yes. With probability 1, there's a theorem that states that with probability 1, this is going to converge to a root. And this root in the population genetics space is called the grand most recent common ancestor. The key word is most recent. In computer science, there's a term called a least common ancestor, or LCA. And the property that LCA has in both this structure and this is that it is ancestral to all the leaf nodes, and none of the nodes here or here have the same property, satisfy the same property. And now if you're in the business of reconstructing common history, you suddenly begin to realize that this structure is very complex. So the question that comes to mind is, can you even reconstruct this history? And we pose this problem as follows. What is the fraction of the common genetic history that is impenetrable? How do you answer this question? I'm going to use random graphs. So I'll use a random graph framework. Here, the nodes are arranged in rows. There is a sense of direction to this. The present or the extant units are at the bottom row, and the past is at the top row. So the genetic material is flowing from past to present, from the top rows to the bottom rows. And every node here, if it is not isolated, has two parents from the previous rows. And that two incoming nodes which correspond to the two parents. And this captures the flow of the genetic material. And now if we are tracking the history of these 4 units, you can see this is the structure that we will get. So this is the arc structure that you will get, and you see that you have nodes. You have some nodes which have multiple parents, like this node here and this node here, and so on. Now, does this completely model the arc? Actually not. So let me show you a simple example. Here are 2 leaf nodes, and this is the LCA of these 2 nodes. But if you look at the genetic material inside, it is not being transmitted to these two units, or these two units did not inherit the nodes from here. And how did that happen? Because recombination is a rather tricky operation. So if I look at the genetic segment here, and if there is a recombination, then part of it comes from one parent and the other part comes from the other parent. Of course, if there was no recombination, the whole thing would come from this parent or this parent. And this leads to what we call the ancestor without ancestry paradox. That is, this is an ancestor, but it has no ancestry. So we need to fix that. So this topology enough is not capturing the arc. You have to associate with rules that capture the flow of genetic material. These are very natural rules that, intuitive natural rules that we associate with this. And now the model is complete. I show you this arc here, which is exactly the same arc that was there in the last slide, except now the edges and the vertices are annotated and the annotation is shown as the colors. And these are the 4 extant units, and these are the SNPs that they have. If you follow these characters down to the leaf nodes, you will find who has these SNPs and so on. So now the next question we ask is, are all these nodes necessary in this? Can I knock off some of these nodes, redundant nodes, without affecting the sample space I have? And some other properties, but let's not go into that. Let's look at only the leaf node samples we have. It turns out I can knock off this node and this node, and then iron out the arc, and I have the same samples, and no other node can be knocked out from here. And that is the notion of a minimal descriptor. We define the concept of a minimal descriptor, and we prove that given any arc, it has a unique minimal descriptor. So you can knock out the nodes in any order as groups and so on. You'll end up with this structure that preserves these samples and certain other properties. So it is preserving the information that is contained in the arc, information that is of interest to us. So now this minimal descriptor becomes an upper bound on any arc that is reconstructed by an algorithm. So now we have the ingredients to answer the question that I had posed before. Is that the first one, I can simulate the arc and then extract the minimal descriptor and compare the sizes. For this, we turn to the community to provide us simulators for populations, because we didn't want to influence that with our own simulators. And the only thing that we ask for from the simulators from the community is that give us access to the arc so we can compute the we can extract the minimal descriptor. We run many replicates, average them, and what we find is that no matter the richness of the sample in a population, at least 1/3 of the population genetic history remains impenetrable for these demographies, and I'm going to assume for most demographies. So moving on to the next problem. Usually people simulate the arc to study certain characteristics. They run tens, hundreds, thousands of thousands of simulations, and then average over these and are looking at the characteristics like what is the height of the arc? How far back in time is the GMRCA? You can look at number of recombinations, number of mutations, diversity in the populations, and so on. For this, we provided a closed form solution to the expected value of these. How could we do that? We use the random graph framework, of course, and then we have compared this with our simulators, external simulators, to match the expected values that we get from here. What this implies is that we have obviated the need to simulate. You don't need to simulate. You don't have to run tens and hundreds and thousands of simulations. If your interest was in these characteristics, you can just give the parameters to these equations and just obtain them. Now again, if this line of research has intrigued you, there is a lot of space for you to contribute. For instance, there are evolutionary processes that modulate the arc, like selection. There could be single loci, multiple loci under selection. They could interact, epistasis. And then under those circumstances, can you still escape the morass of simulations? Now I move to the second part of my talk, which I call big data genomics. And your data could lie on a point cloud in a coordinate space, or your data could be in a coordinate-free space, in what I call a relation space. For example, if your input data is a network, you're capturing the relation between these two nodes. So one dimension to this is, of course, the number of points, or the number of points, the number of edges here, and the other dimension, which is literally the dimension, is the dimension of this space. And if you have been working in this space in statistical learning, you have probably faced the wrath of dimension, the curse of dimensionality, which actually afflicts both these spaces. This even much more than this. This is the dimensionality is on steroids here. But I'll first cover a problem in this. I'm calling this statistical learning, and this is AI and machine learning methods that you're probably familiar with. I'll use an example here. Many of you may know that my team in IBM Research, we worked on cancer genomics clinical decision support system, which is also now an IBM offering. And we continue to work in this space. We are working on a project of cancer drug resistance with Broad. Gad Getz is the PI, and I believe he's in the audience somewhere here at ISMB. And the next problem I choose from this space of cancer genomics. And the question we ask here is the disease subtype signal in the genome? Literally in the genome, in the DNA. We're not going to look at RNA, we are not looking at proteomics data, we are not looking at images or any other accompanying data. We just want to look at the genome and see whether we can get the subtype signal from there. We're looking at whole genome sequencing data, about 727 patients, and 7 forms of hematological cancer, AML, BALL, and so on, shown here. Why is this problem difficult or interesting? Because these diseases, the origin of the disease is in a few cells developing from the hematopoietic stem cells, and that makes the problem a little difficult. So we use machine learning and AI. What I show you here are these 700 odd patients, and the color of the dot is the subtype. You can see that it's not separating well. We try very hard, and it's better than this, but it still does not separate. To cut a long story short, we introduce what we call stochastic regularization of the data, which is a 2-step process. So what this does is it takes a feature and disperses it into a distribution. And it has a 2-step process. The first step is outside this replicate cycle, and the second step is inside, hence stochastic. And when we use this method, it looks like we can separate the diseases. So indeed, there is signal in the DNA to separate these cancer subtypes. The next natural question is, where on the genome are the signals? So what we do, what I showed you in the last slide was the result of whole genome. So we divvy the genome up into genic regions, non-genic regions. The genic regions are divided up into exonic region, intronic region. From the non-genic region, we take annotated regions. And whatever is left is the dark region. If we know anything at all about that, it is pulled into the annotated region. And these 2 concentric circles are showing you that these regions are distributed throughout the chromosome, all the way going from chromosome 1 to chromosome 22. And now what we will do is, we will look at these regions separately and see how they separated the disease subtypes. Now, I show that to you in terms of colors. The darker the green, the better it is. The lighter the color, the worse it is. In the bottom row, you are seeing permuted data as a quick sanity check, where you permute the labels of the disease, not the genome. You don't touch the DNA data. You just permute the subtypes. And this should be light, and it is indeed light. And this is the original data, and this is sort of an average of off-the-shelf AI methods. And you can see that it is somewhere in this range, kind of mediocre. And then we use the stochastic regularization method, and you see that this gets darker and this gets lighter, suggesting that perhaps we are doing something right. And it has different values for these different regions corresponding to that, but overall it is separating them well. But what perhaps is not clear from this picture is that this region, which corresponds to the dark region here, does the best in separating the signals, suggesting that dark matter matters. And we also did some sanity check by normalizing these sizes because they are of different sizes, but the results continue to hold. So the takeaway from this is that, you know, an appeal to the community to spend time, energy, money on exploring dark matter, because it appears that perhaps they carry some information that is of consequence to the applications that one looks at. So now I move to the next class of problems, that of the coordinate-free space or the relation space. And I will use a tool called topological data analysis, or TDA. I'm going to assume that most of you are not familiar with TDA. So I will take the liberty of giving a small primer on this so that I continue to be intelligible to all of you. So let's start with something that you are familiar with. So this is a graph with 11 vertices and no edges. And so V is equal to 11. And in the language of algebraic topology, this is the 0th Betti number, which corresponds to the number of connected components, which is 11. So I'll show you these values, or the summary of your graph in 2 columns. And let's proceed to now time t2, where I add 7 edges to these points that I had. So now I have 11 vertices and 7 edges. And the 0th Betti number now is 4, because we have 4 connected components. So now you can recall from your undergrad or grad classes that the alternating sum of this, which is 11 minus 7 equal to 4, which is the Euler characteristic, corresponds to the number of connected components here. So we'll take the alternating sum on this side and alternating sum on this side, and they match, and this is 4. So now let's move to time t3. I'm saying time, but it can be any other parameter. I add 2 more edges. I add this edge and this edge here. And now the alternating sum becomes 11 and 9. 11 minus 9, 2, but I still have 4 connected components. So what happened? What happened here is that we introduced 2 cycles here, and that's the first Betti number is 2. So these are 2 holes or 2 cycles, and we have to now take the alternating sum of this, and they still match. So an important point to remember here, that in topology, holes matter. A hole is an important characteristic. I will now move to the next time point. And here I'm adding a hyperedge between these 3. So here are these 3 points. This hyperedge is shown in blue. That is, it is an edge or hyperedge between 3 nodes instead of the usual 2 nodes. And that's a 2 simplex. And then what happens is that this particular cycle that we had here vanishes, because now it becomes a boundary, okay? But I also added 2 edges here. So I added 2 edges here, and the first Betti number, which is 3 here, should correspond to 3 cycles. But if you are seeing more than 3 cycles in this space, you are seeing right. So why is it 3? It is 3 because 3 is the dimension of the vector space of the homology cycles. That is, all the cycles are not independent. The number of independent cycles is 3. And this is another point you might want to keep in mind, is that 3 is the dimension of the cardinality of the basis, but the basis is not unique. But we might need to map, go from here to our input data. So there's another point we might want to keep in mind. So for the sake of completeness, let me add another time point here, and I'm adding 4 more 2 simplices. So imagine here is like a tetrahedron, and you have added the 4 faces of the tetrahedron. So the 2 simplex becomes 1, and now you lose all the first Betti numbers, no more cycles, and you have a hole in the next dimension. So the second Betti number is 1. And again, the alternating sum on the left matches the alternating sum on the right. So these are 2 alternative ways of summarizing the structure that you have. But of course, these holes, there's a little bit of more detail to it, and this is called a bar diagram here. And the bar diagram, you can see, let's follow B0, B1, and B2. B0, imagine that you're seeing 11 lines here, and then you see 4 lines till the end of time. So B0 was 4 throughout. And then B1, you have 2 lines here, and then 3 lines here. A little bit of detail here because one of them vanished. So one was born and it died here because this was the hole that was born here and it died here. So you don't see that bar anymore. Instead, one of the bar continues and you have 2 more bars here, and this 3 continues to be. So you have 3 bars here because B1 is 3. And B2 is 1 born here and continues till the end of time. So this is the this is the topological summary, and it summarizes as we move from, say, this time point to this time point. So let me summarize what are the components here. First of all, I have a simplicial complex. I did not use that term here, but it refers to the graph structure that you had here. This graph is a simplicial complex. So is this graph. Anything that is lying on a top mat, which you know as a graph, is a simplicial complex because you know you're adding higher dimension simplices as you go. You can have different types of simplicial complexes. And the next component, the next element we saw here was the filtration of the simplicial complexes. That is, as I moved from time T1 to T2 to T3 and so on, I was adding simplices as I went, not removing, but adding. So this is contained in this, is contained in this, is contained in this, is contained in this. So it is a chain of containment, and that's called filtration of the simplicial complexes. And then you saw the summary in terms of, here we see them in terms of bar diagrams, but essentially what you're seeing is the birth and death of homologies or these cycles. Now we have these 3 steps and we can apply it to our problem. So let me give you a quick example of a problem where we literally used this notion of homology or holes to solve a problem. And that is of admixture in populations. So you remember this, this structure that I had shown you at the beginning of the talk. This is capturing the common history, recombining history. So it is peppered with cycles because of recombinations. And if we take a 30,000 feet view of this, say, this is what it looked like. And so let me take a step back and try to define what is admixture here. Is that A, C, D, B, these are the populations that you are seeing. And of course, the detail that you see here corresponds to the structure here. This population is admixed because it is a mixture of this population and this population. This is admixed because it is a mixture of this and this, or interbreeding of these 2 populations, while A and B are not admixed. And if you pull out a scaffold, if you will, from this, you see that you get these 2 big cycles here. And these 2 big cycles you see associate in a way with the 2 admixtures that you're seeing here. So why is this problem tricky or even interesting is that we know that this arc is peppered with cycles because of recombination. So there are small cycles everywhere. Now the question is, can one separate these big cycles from these small cycles? We proved a theorem which says that highly persistent homology meaning the bars which are very long of the simplicial complex of this arc is approximated by the persistent homology of the simplicial complex of this one. So this gives us hope that we can separate the small cycles from the large cycles, but of course in practice you have only the distance matrix or distance between the leaf nodes, and from that you can possibly deduce recombination. We ran this on simulated data, complex scenarios of simulation of admixture, and we assured ourselves that we could separate the admixture, admixed population from the non-admixed population. The data here is from our collaborators in USDA on avocados, and we use cultivars which were admixed, which were not admixed, and using TDA we were able to detect the admixed cultivars. Now I want to move on to another, rendition of TDA, if you will, for a slightly different kind of problem. So again, many of you may know that we have been working on microbiome data in the context of sequencing the food supply chain. We are working on AI for healthy living with human microbiomes. This is work with Rob Knight. And so the next problem I pick from this space of metagenomics. So in the interest of time, I'll give you a quick description of what the problem is. And here, and I'm focusing on the signal enrichment of the NGS reads. The problem is as follows, that you have multiple reads from a sample, and you are going to map them to reference genomes in a database. So why is the problem interesting or tricky? Because these reads are promiscuous. They map to multiple references in your database, either because the reads are small or because the references are close to each other, references are very similar, whatever it is, the reads are promiscuous. And so what is the traditional approach to this problem? That is, you focus only on those reads that map uniquely to a reference genome, and then either you discard the promiscuous ones or take the promiscuous ones with some reduced weight. So we set out to use TDA to solve this problem, if we could. So this is a simple example. So you have, say, 3 references. You have reads that map to A and map to B. You have reads that map to A, B, and C, and unique reads and so on. And there is a natural Venn diagram that you can construct from here. And this corresponds to reads that uniquely map to these genomes. And this corresponds to reads that map to both A and B, and this corresponds to those reads that map to A, B, and C. So I had earlier talked about a relation space, right? So this is a relation between A and B. This is a relation between itself, A and A, and this is a relation between A, B, and C. And in practice, we see that this relation space goes up to 20 or 30 in the kind of data that we have been looking at. And this is what I meant by the relation space. And this is what we'll be using to use a TDA here. And for people who are already familiar with this problem and genome and so on, of course, I'm showing A, B, and C as of having the same length. They're not necessarily of the same length. And there is another piece of information, and that is, what is the coverage or depth of these reads in each of these? So for example, if I take some entry, if I take these entries in AB here, I can get the coverage in A, or I can get a coverage in B. So I will have these multiple numbers. Clearly ABC are not of the same length. If they're of the same length, this could be the same. So we have this added information. So these added information is associated with this Venn diagram here. And now we have the components of TDA. There's a technical point here. I don't have to get into it. To make the filtration possible, we had to use a construct called barycentric subdivision. And after that, we're able to do the filtration. And so here, what is the filtration? So there is no notion of time. We do the filtration by the coverage. So as you increase the value of the coverage value, the coverage D values that I showed you here, you begin to get these chain of containments. And then there is the step of essential simplices. Again, at the beginning, I had said that since the basis is not unique and we want to interpret our results, we must have a way of going from the bar diagrams to our input. So we define a notion of essential simplices, which enables us to do a unique way of associating the input to the bars. And then using this result of the filtration that I had explained and this notion of essential simplices, we are able to score the OTUs or the organisms that we have. And when we run them on simulated data, we find that the true positives do bubble up to the top as we wanted them to. But I had a curiosity, and we wanted to use this without genome coverage. So so we had this extra information. This is of course part of the problem, and we were happy to use that information. But what if I didn't use that information? That is, I don't care what is the depth of these reads or what is the coverage on the genome. I don't care what are the size of the reads. All I care about is these numbers, just the number of reads. So I will just have these numbers, and now can I do TDA on this, and what results will I get? So we go through the process. We can associate what is called a check complex. This simplicial complex is called a check complex to this structure, and then we crank it through our TDA steps. We do the filtration. Now here, the filtration is by that number of reads, these numbers. So these numbers are your time. So that's the one that gives you that chain of containment. Of course, we need essential simplices because we want to go from these bars to our input, and then we score them. A natural way of scoring them based on this filtration and essential simplices. And to our great surprise, it actually worked. In most cases, it worked even without having the depth information. In simulated cases, in real data and so on, it was working fairly well. So here I'm going to cheat a little bit, and I will show you one of the best case scenarios. Here, the green ones are the true positives that we knew from the experiment. You can see they have bubbled to the top. The blue ones were the top entries for a method that did not use TDA. So we find that in a slim model like this, we actually use less information that the problem has, yet we are able to answer the question. And it even outperformed the traditional way of solving this problem. And next, I want to show you an example of combining these two methods. And you can ask, is your data lying in the coordinate space, or is it lying in the coordinate free space? It's a good question. And I will say space lies in the eyes of the beholder. So let me explain what I mean. So here we use a Parkinson's disease data from MGFF, and simple problem. We have case control data, and we are looking at the gene expression, and we used TDA in combination with AI to solve this problem. Let me walk you through this process. So on the training samples, we extracted what we call a gene skeleton, which is actually based on a correlation distance. This is just a schematic. We don't have to take it literally. And then we individualize these skeletons by assigning or associating the gene expression value of that subject. Of course, these expression values are appropriately normalized. So now what it means here is that imagine you have a ball on each of these nodes which correspond to the genes, and each ball is of corresponds to the gene expression value of those genes, and you expand the ball as we go, and that's our containment filtration that we are doing. And the expansion is at the rate that is determined by the expression value of that gene. And as you expand them, the different balls begin to collide or overlap, and you begin to get a relationship between 2 balls, 3 balls, 4 balls, and so on, taking us back to the relation space that I had spoken about before. So now we can do TDA on this. I told you what is the filtration. For the filtration, the knob for the filtration is the expression value, the rate at which we are expanding this. Then we can get the topological summaries. And now these topological summaries are fed into your AI learning. And we find to our surprise that the combination of TDA and AI method actually improved the results from just using AI on that. In most cases, maybe except for this, but in most of the measures that we used here, we found that there is an improvement here. The toolkits for AI and machine learning probably are more ubiquitous than TDA. So we are working on a multipurpose toolkit for TDA called Matilda. These are the overarching principles for how we handle explosion, how we handle, the cursive dimension, or and some other technical details like coefficients in any finite field and so on. It can be used as a library, or it can be used as a standalone, and we offer a comprehensive combination of input and output, essential simplices. And here's something we're dabbling with on the SARS-CoV-2 on one kind of mapper or one kind of output here. This toolkit is under construction, and once it's ready, we will share it with the research community. So to recap, I showed you that you can use some combinatorial structures and algorithms to get the most efficient algorithm possible in gene clusters, with interesting spill out in computer security and even classical mathematics. And we used random graphs and statistics to answer the ARG reconstructibility problem, and also gave you a way of escaping the morass of simulations, if you will, at least in some cases. We used AI to show that, to uncover that even the dark matter part of the DNA perhaps plays some role. And in metagenomics, I showed you that even a slim model not just answered the question of signal enrichment, it even outperformed the traditional approach. Of course, the idea here is not to supplant other methods, but to get them to work in synergy. In Parkinson's disease, I showed you that combining machine learning and AI actually improved the improved the results of AI. So I hope I've convinced you that mixing and matching methods and drawing from these different techniques and from different vantage points is not just fun, it's also useful. And all the work that I showed you today spans many years, many external collaborators, and many papers. And you will find all these papers here, or if you're able to scan, you can just scan this code. So I would like to acknowledge and thank all my collaborators and their entire team, which made all the work possible. In particular, I would like to thank Joma for the ARG reconstruction question, Arun for the computer security question, David for the plant genomics question in Admixture, Bart for the microbiome problem in food safety, and Torsten for the hematological cancer. Finally, last but not least, my team, without whose diligence and dedication, none of these projects would have either taken off or even landed. Finally, I want to share this with you, that in response to COVID-19, IBM is sharing many of its resources, making it available for the research community, for the community at large, at no obligation, no cost. Here is a list of 3 resources, and in this URL, you will find the collection of all the resources, an ever-growing collection of resources here. And I hope you'll check them out. So with this, I will stop. Thank you for your patience and time. And I welcome any questions you may have, and I hand it over to David for that.
Okay, thank you, Laxmi. So the first question is, I'm not sure how mitochondrial genomics apply to the recombination generated paradox of ancestry without ancestors. Is there recombination in mitogenomes, or did I misunderstand something?
No, so the mitochondrial DNA would only give you a tree. But if you want to look at the history of a recombining segment, any of the other autosomes, it will be an arc. So the mitochondrial DNA will only give you a tree. So that's why the figure that I showed you for the mitochondrial DNA was a tree, and that was a correct tree. Yeah.
So second question, does dark matter include common repetitive elements?
If it has no, and if it is not, doesn't have any other annotations to it, we have included that. If it has annotations, it gets into the annotated regions. So this is something we can take offline and discuss, because there's a lot of details on what was included in the annotated region. So I can take that offline, or I can let me go to that particular, yeah. So the annotated region here is pseudogenes, non-coding RNA, transcription, factor binding sites, enhancers, promoters, and 5' and 3' UTR. And this is more or less the complete list, but we can discuss if there were others that was included here.
So, for example, lines, okay, were they eliminated from dark matter? I guess if they're annotated, they're...
Yeah, these are all good questions, but they are a little more technical and detailed. I think we can take a closer look. We can chat offline and discuss
that. So, a question, one person says, my assumption is that dark matter would be under for less purifying selection. So that we were looking at information sets that separate recently evolved cancer lines, would we not expect this portion of the genome to contain more information for classification? How does this relate to what matters for cancer?
Okay, I didn't understand the whole question, but the point here is we are just using a sort of a black box method of AI, right? And we are saying that we don't know the mechanisms. We don't know what it is. The question we ask, so you see, the question that we ask is there a signal in the genome or not? Okay, and then we take this and use a black box method to see if it is separating. And if it is separating here, our conclusion from there is possibly there is some signal, there is something happening there. We don't know what it is. We don't know the mechanism. It probably needs some more exploration there.
And we have a more general question. What are your thoughts on the future directions of bioinformatics?
That's a very good question, and a broad question. So I personally would, there are many directions in bioinformatics can go based on your expertise and your views and your thoughts. I would try to take it in the direction where we are bringing in more techniques and other things that we learn from mathematics, computer science, kind of combining them, trying to answer more and more interesting and more difficult questions as we go. And that's the direction I like the field to go in. I would like the field to go in.
Another question about dark matter, which I don't know, but they understand, but the question is it possible that the dark matter is in LD with non-dark matter?
To this, I will say no, because the region is very large. Again, we can take a closer look at this, because the dark matter region is fairly large. And so what is non-dark matter, for example? So let me just, okay. So the dark matter is pretty large to be in LD consistently with other non-dark. So the short answer to that question is I don't think so they are in LD. Although we didn't look at it, but I doubt that such a large region is in LD with them. So to that I will say no.
Including the information from the gene expression data and TDA, will we be able to decipher the functioning of certain conservative genes?
I hope we can. We will probably have to explore this, and I think this is a fun space to bring in a slightly a different view and vantage point, or different how you are looking at the data and what you are how you are posing the question and you're getting the answer. I think it's a fun space for us to play in and see if it answers some of the difficult questions. And not to say that this is the only way. No, there are a lot of other methods dominated by statistics and so on, which is very good, and people, we buy those answers much more than we probably buy some indirect methods. But statistical learning is here. We take the answers without understanding how they did it, and of course we are now exploring and getting explainable AI and so on. So this will move in that space. I do believe that bringing in these independent, very different tools will help us answer hard questions.
We have a question. Could this be contribute to different mutational signatures in different cancers?
I'm sorry, I probably didn't get the question. This is in the follow-up to some other question?
Perhaps. I don't know what this is. Could this be contributing to different mutational signatures in different cancers?
Okay, so I'm assuming that this is probably related to the dark matter question. I think that's probably a fairly popular topic. And so here the problem was difficult or interesting because it is the subtype of the same disease, right? So that's why the problem becomes challenging, because we believe that if it were a gross type of, like kidney cancer and pancreatic cancer, breast cancer, probably the signature would be much clearer. And this is a more difficult, nuanced problem. And so we went with the subtyping of one kind of cancer. All of them originating in the hematopoietic stem cells. So this was hematological cancer.
In the metagenomics read assignment problem, is it interesting to allow read mapping to internal nodes of the species tree? Does it make the problem easier or more difficult?
It's a good question, and that is another aspect of the problem where, in fact, the mappings, we didn't spell it out here, but the mappings do go into the internal nodes. Not all of them map onto a leaf node of your internal taxonomy. They will go into the internal higher level nodes, but that was not an aspect that was explored here. And that's not the dimension that we have seen here, but it's a good point that one can even try to bring in that angle and see what difference it makes. Here, by default, we're assuming that we would probably, in some cases, even assign it to the internal nodes. I guess your question is coming from the fact that if it is mapping onto multiple references, you should probably look at a common ancestor there. That's a good point. But we can't universally do it, because eventually when we want to answer what lies in your data, say in the context of food safety, we probably want to identify the organism as specifically as we can. So there is a balance between can I climb up the taxonomy tree or deal with promiscuity, and that's a balance we have to play.
What advice would you give to biologists who aspire to get into the bioinformatics field?
I have a personal bias, right? And I think the bioinformatics field, I'm going to assume, is with when you're bringing in techniques, mathematical techniques and such to answer questions in biology is very interesting. And the advice I would give to biologists if they want to get into this field is, sure, give it a try. And there are many books, courses, and other things, and it's possible. I find it very exciting, and I'm sure if you have that inclination, you will find it exciting too.
I think that sums up the questions. And I just want to thank Laxmi for her work and for continuing to open new fields and think of things outside the box, and putting things together that are in different boxes, and continuing to amaze everybody. And nice to see you.